Welcome to Lab 4, Competition and Natural Selection. Before we go through the lab, let's highlight the main concepts. What are the conditions necessary for evolution by natural selection to occur within a population? What is fitness? What is relative fitness? What are ways to estimate reproductive success? What is the value of a statistical analysis?
First, you should be familiar with the conditions necessary for a population to evolve through the process of natural selection. Let's go through them. 1. Within a population, there must be phenotypic variation. 2. The phenotype of interest must have a genetic basis, at least in part. 3. Some phenotypes will have a higher probability of reproducing than other phenotypes. You need to understand these and be able to apply them to real-world scenarios. The focus of natural selection is all about the fitness of different phenotypes. But what is fitness? How would you define fitness if asked on an exam? Today, we are interested in relative fitness, or the idea of how much an organism is reproducing relative to others in the population. Why do you think that is important if a population is to evolve through the process of natural selection? Think about this and ask your TA if you still have questions.
In this lab, you estimate reproductive success using components of fitness. This is a way to estimate reproductive success without having to pollinate the plants we are using, let them go to seed, collect the seeds, plant the seeds, wait for those to grow, and then see how many of those reproduce. Now, the big question we are asking revolves around the third condition of natural selection. Is there a difference in reproductive success among different phenotypes? In the context of the lab, we are asking, does competition influence reproductive success? As you do this lab, think about what you learned in Lab 2 about resources plants need to use as well. Also think about ideas from the population growth portion of the course. Most populations exhibit logistic population growth. Why don't populations seem to grow infinitely large?
At home, you have grown Raphanus sativus, or radishes. At this time, I would like to remind you to not consume these radishes as they are lab materials. There are two color morphs, a white morph and a purple morph. The color morphs are based on the colors of their roots, but the purple coloration in the purple morph can also be seen in the leaves. You have grown these plants under different types of competition, which we call treatments. Treatment 1. A single plant of each color morph is grown separately. We call these single white and single purple. As you can see, there is no competition in this treatment. Treatment 2. We planted one white plant with two white plants. In another container, we planted one purple plant with two purple plants. This is our test of intramorph competition. Treatment 3. We planted one white plant with two purple plants and one purple plant with two white plants. This is our test of intermorph competition. Your mission is to determine, is there a difference in reproductive success for two different morphs of Raphanus under different types of competition? This is essentially our working hypothesis. Now let's look at how you will tackle the question. Step 1. First, pick 3 components of fitness to measure. Recall from the pre-lab, you suggested different traits in a plant one can measure. For example, number of flowers or leaf area, and you described how those would influence reproductive success. For this lab, we have picked the components of fitness for you to measure: plant height, number of leaves, and average leaf length. Please note, the plants in the following section were grown differently, but the method of measuring is the same. To measure the plant height, measure from the top of the pellet to the top of the terminal leaf node. To measure number of leaves, simply count them. For average leaf length, measure each leaf and then calculate the average length. For this exercise, you may sample destructively. And remember, please use the appropriate metric units. Step 2. Next, you will measure these traits for each of your treatments. In treatment 1, you will measure all 3 components of fitness for the single white and the single purple. Then, record those data. For treatment 2, the intramorph competition treatment, randomly pick one white and one purple plant to measure for all 3 components of fitness. For treatment 3, the intermorph treatment, measure all 3 components for the white plant grown with the purple plants, and then do the same for the purple plant grown with the white plants. After you have written down that data, enter it into the Google form. We do realize that growing your plants may not have gone well for you. For any plants that you don't have data for, simply leave those parts of the Google form blank. Do not enter a 0. If none of your plants grew, or you did not grow any plants, please check the appropriate boxes. This helps us plan for future quarters. Your TA will convert the Google form into a usable spreadsheet of data. In the next video, we will orient you to the spreadsheet and show you how to do a t-test. That's it for this video. Make sure to check out the next video on how to do t-tests. As always, if you have any questions, please contact your TA.